Today we will talk about the cellular level. So we'll talk about the cells. First we'll see the definition of a cell, the parts of a typical cell. Then we'll talk about the cell membrane, structure and functions of the cell membrane, properties. If you see the structure of a cell membrane, you have two things mainly: phospholipid and proteins. So first we will talk about the proteins on the cell membrane and their functions. We will talk about the intercellular junctions or joinings. Intercellular junctions attach the cells to each other, hold the cells together. So we'll see what are different types of cell junctions that work in our body. Then we'll talk about the transport across the cell membrane. Cell membrane is a semi-permeable membrane. That means it selectively allows certain chemicals to pass through it. So we'll see the processes of transport across the cell membrane, how the chemicals pass through the cell membrane. Then we'll talk about the response of a living cell in three different types of solution: hypotonic, hypertonic, and isotonic. So if you put a living cell in those three types of solution, what kind of change you will see in the cell. Okay, so mostly we'll talk about those things. First, what is a cell? A cell is the structural and functional unit of the body. Human body has 50 to 100 trillion cells, not million, not billion, trillion of cells, and over 200 different types of cells are present in the human body. How they are different? They are different in size, shape, subcellular components, and functions. So that's how they are different from each other. But one thing is common for all the cells. All cells consist of or composed of chiefly CH, N, and O. That means carbon, hydrogen, nitrogen, and oxygen. You must remember from last class I said that about 96% body mass is formed by these four elements, right? 96%. So these are the main. Here just you see how different cells are different from each other. A few examples. First, if you see a fibroblast, fibroblasts are long cells and they produce fibers, connective tissue fibers. Okay? So, fibroblasts are elongated, thin, and they produce fibers. They are connective tissue cells. Erythrocytes are round, flat, round but flat, and The color is red. Erythrocyte or red blood cell color is red, and function is transport of oxygen. Cuboidal epithelial cells are cuboidal in shape. So if you see cuboidal epithelial cells, they are cubical shaped cells, like cubes. Okay? So these are the cuboidal epithelial cells. Cuboidal epithelial cells cover the linings. All epithelial cells mostly cover the linings, form the linings. Muscle cells. If you see a skeletal muscle, it is very long and cylindrical, and multiple nuclei are present inside the fiber. So fiber is very long, cylindrical with multiple nucleus. But if you see smooth muscle, it is different. The smooth muscle cells are not cylindrical but spindle shaped, and only one nucleus is present in the center. So you see, even different types of muscle cells are different from each other, right? In shape, size, and structure. Fat cell. If you see a fat cell, the fat cell is round, but nucleus is not in the center. Nucleus is found attached to the membrane, the side of the membrane, like this. Okay? And neural cell, if you see a nerve cell or a neural cell, it has a cell body and processes, a number of processes. So you see in the right top, a neural cell has processes, dendrites and axon. Now the function of a neural or nerve cell is electrical signal transmission. So it's completely different than other cells. Electrical signal transmission. Sex cell, if you see the male sex cell, that is the sperm, it looks like a baby fish. It has a head, body and tail. Okay? Why its structure is like that? Because it swims in the fluid. That's why it needs a tail. By beating the tail, it moves. So you see just how different body cells are different in shape, size and function. There are more than 200 different types. Now, the parts of a typical cell. A typical cell has three parts. Cell membrane, which is also called plasma membrane, that gives the boundary of the cell, and then you have cytoplasm. Cytoplasm is the part between the cell membrane and the nucleus. So this is the cell, this is the nucleus, so this is the cell membrane or plasma membrane. This is the nucleus, and the fluid between the plasma membrane and the nucleus is the cytoplasm. In cytoplasm, you have different types of organelles. So organelles are present in the cytoplasm. We'll talk about those organelles, okay? Float in the cytoplasm. Plasma membrane, as I have mentioned, gives the boundary, and Cytoplasm contains organelles. Two types of organelles are present in the cytoplasm. So this is the cytoplasm. Two types of organelles are present in the cytoplasm: membranous and non-membranous. Then in the center, usually in the center, you'll see in nucleus. Nucleus is the control center. Why? Because in the nucleus you have the chromosomes, and in the chromosomes you have the DNA. DNA controls the functions of the cell. So DNA are located or present inside the nucleus. That's why nucleus is called the control center of the cell. So what are the membranous and non-membranous organelles? I'll just mention few. You have learned definitely about the cell in your high school and intro bio, right? So we'll quickly go over, not in detail. So important organelles in the cytoplasm are mitochondria, endoplasmic reticulum or ER, Golgi apparatus, ribosomes, vesicles. So those are important, some important organelles. Mitochondria, endoplasmic reticulum, Golgi apparatus, ribosomes, right? Vesicles are also called, here let's see, lysosome. Same thing. Okay? Vesicles are round structures, membranous structures filled with chemicals. Lysosome or vesicles. There are more, just we'll talk about those few. First, mitochondria. Mitochondria are the powerhouses. Why they are called the powerhouses of the cell? Because they produce ATP, and you know that ATP gives energy. We have talked about that in last class, right? So mitochondria mostly produce ATP. That's why they are called the powerhouses of the cell. Endoplasmic reticulum or ER has two parts: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough and smooth ER. Why one part is called rough and another part is called smooth? Because if you see the endoplasmic reticulum, it looks like many tube-like structures are connected to each other. Many tube-like structures are connected to each other. Network of tubes. And one part of ER has ribosomes attached on the surface. So these are the ribosomes attached on the surface of the endoplasmic reticulum. This part is rough because surface is rough, and this part is smooth. No ribosomes attached on the surface. We also use the term rough surfaced ER, smooth surfaced ER. Now, the function of ribosomes. Ribosomes are these tiny particles. Ribosomes manufactures proteins. So, synthesis of protein or manufacturing of protein is the function of the ribosomes. Synthesis of protein. Then that protein enters into the ER. So ribosomes produce or manufacture proteins, then that proteins go inside the ER. And rough ER does what? Rough ER processes proteins. So that proteins are processed after manufacturing, that proteins are processed inside the rough part of ER. Okay. Then the proteins are sent to Golgi apparatus. So from here proteins go to Golgi apparatus. If you see the Golgi apparatus, they look like many flattened sacs are piled together. So these are the flattened sacs are stuck together. That is the Golgi apparatus. So proteins, after processing of the proteins, rough ER sends the processed proteins to the Golgi apparatus. Okay. So now proteins are here. These are already processed, and Golgi apparatus does the packaging of the proteins. So packaging of proteins. Is the function of the Golgi apparatus. So you see, it's like a factory, right? We know if you go to a factory, it has different sections, right? Production or manufacturing, one section. Then processing, folding. You can also add folding. Folding of proteins, okay? Then sent to the section, they will do the packaging part. So very similar to a factory where they manufacture, process, and do the packaging to send them out. Make sense? So those are important functions of ribosomes, ER, rough ER, and Golgi apparatus, and they deal with proteins. Now, Golgi apparatus sends the protein molecules to the ends of those sacs, flattened sacs. So from the Golgi apparatus, the proteins mostly accumulate at the both ends of the sacs, like this, okay? And then what happens? The ends get detached from the main part, like this. They get separated, both ends. After the protein molecules accumulate, they get separated and form lysosome or vesicles. Okay, lysosome or vesicles. And now the vesicles have the proteins inside. Vesicles move to the cell membrane or plasma membrane. So vesicles will go and get attached to the cell membrane or plasma membrane, and will get what? Rupture to release the chemicals to the outside of the cell. Okay, so vesicles, you see in that picture, vesicles will get attached to the cell membrane and rupture to release the chemicals to the outside of the cell by exocytosis is the process how the vesicles or lysosome release the chemicals. That process of rupture and releasing is called exocytosis. Exocytosis, sending outside. Okay, now smooth part of ear. This part is the smooth ear. The function of smooth ear is number one, lipid synthesis, synthesis of lipids or fats. Number two, glucose metabolism. Number three, detoxification, another very important function. Detoxification is an important function of a smooth ear. You know that metabolism produces harmful toxic chemicals, right? Cellular metabolism produces toxic chemicals in our body, and also we take the food all the time and some toxic chemicals are going inside, right? When you eat some food has good nutrients as well as toxic chemicals. Some of those toxic chemicals are detoxified, neutralized or inactivated in the lysol. Makes sense? The important function in our body. So just know those few functions. We won't talk to detail because you have already learned about the cells in previous classes, definitely in intro bio and high school. Plasma membrane or cell membrane is a very important part of the cell because it gives the boundary of the cell. Why cell needs boundary? Because intercellular fluid must be separated from extracellular fluid. So by providing boundary or giving boundary, cell membrane separates inside the cell from outside and maintains internal environment of the cell. Cell membrane, if you see the structure of a cell membrane, in one sentence you can say cell membrane is a phospholipid bilayer in which proteins are embedded. So cell membrane is a phospholipid bilayer in which proteins are embedded. Now, why we say phospholipid bilayer? Because if you see the structure of a cell membrane, it has two layers of phospholipid. That's why called phospholipid bilayer. These are the phospholipid molecules. So, phospholipid molecules form two layers. This is one layer of phospholipid. This is another layer of phospholipid. Okay, and in phospholipid you will see proteins. Often proteins are embedded. So this is the structure of the cell membrane. Now, this is a layer of phospholipid, this is another layer of phospholipid, and these are phospholipid molecules. So if you see one phospholipid, it has a head part and a tail portion. The head portion and tail portion or parts. Head part is also called polar end, and tail part is also called non-polar. Head part loves water, likes water. That's why this water liking head part is called hydro. Hydro means water, you know that. Water, hydrophilic part. And tail part dislikes water. That's why this part is called hydrophobic. Hydrophobic is the tail portion. So you see, now I can tell this entire part doesn't like water. Does not like water. Only the outer and inner surfaces like water, but entire middle portion doesn't like water. So water molecules cannot easily pass through the phospholipid bilayer. Water cannot pass through phospholipid bilayer because this part doesn't like water. Okay. Now these are proteins. Okay. Two types of proteins are present in the cell membrane. Some proteins are deeply inserted in phospholipid bilayer. For example, this one is going all through, passing all through the phospholipid bilayer. This one, although not going out through, but it is deeply inserted. So these are called integral proteins. Integral proteins. Okay. Some proteins are loosely attached to the integral proteins. They are not inserted in phospholipid. So these are called peripheral proteins. Peripheral proteins. And these are integral proteins. Okay. This is also integral. Integral protein. Now we'll see the functions of these proteins, okay, in the cell membrane. Both integral and peripheral proteins have some important functions. We just mentioned a few of those. Integral proteins help in transporting the molecules by forming channels and working as a carrier. So integral proteins help in transport. Some integral proteins act as enzymes, and some integral proteins act as receptors. So transport, acting as enzyme, and acting as receptors. Those are main functions of the integral proteins. We will see how they perform those functions. Peripheral proteins, just write down two important functions I will mention. One is peripheral proteins form intercellular junctions. Intercellular junctions hold the cells together. So if you see the cells, for example, these are epithelial cells. Peripheral proteins form intercellular junctions and those junctions mainly are used to hold the cells together. We'll talk about different types of intercellular junctions, just know that. That is one function of peripheral proteins. Another is cell-to-cell recognition. Cell-to-cell recognition. The cells of your body can identify the other cells. That ability is the cell-to-cell recognition. You know, when your body receives tissue from another person, most of the cases what happens if you just do tissue transplantation? Rejection, right? Because your body cells can identify if the cells are coming from your body or from another person. Make sense? So that is cell-to-cell recognition. But if I take tissue from skin from another part of your body and put, it will accept, right? So again, cells know that coming from your own body. So that is a big issue in transplantation. Immune response occurs, right? So cell-to-cell recognition is given by the peripheral proteins, very important function. Here you see the structure of a cell membrane, mostly phospholipid bilayer, in which the proteins are embedded. You have a small amount of cholesterol or lipid and glucose. In this picture, the green chains are glucose and the yellow chains are fatty acids or fats or cholesterol. Okay, so those are present in a small amount. How the integral proteins help in transportation of chemicals. In some integral proteins, you will see the channels, through channel. So the molecules smaller than the size of this channel will easily get in or out. So smaller than the size of the channel, those molecules will easily get in and out, right? And these channels are always open. Some integral proteins have channels, but those channels are gated channels, remain closed. To open those gates, you need what? ATP. Make sense? To open the door, you need energy. So that's how the channels are formed by the integral proteins. Some gated channels, some open channels. Now, to open the gates, you need ATP. How the integral proteins work as receptors. Some integral proteins, so just think this is the cell membrane, this is the integral protein, okay? This is another integral protein here, this is the cell membrane. So, in the outer part of the integral protein, in some integral protein, you have binding sites for the chemicals. For example, in this integral protein, you have a binding site, and the shape of the binding site is like this. On this one, you have a binding site, but the shape is round like this. Another one you have here, the shape is, or say, square or rectangular. Okay. Now, if you take a medicine, for example, you have taken a medicine, and the molecules are round, like this. This is the signal molecule. Signal. It could be medicine or hormones in your body, right? So drugs go from outside, hormones come from glands. Those are the signal molecules that work on the cells. So this one will nicely hit here. Make sense? Because round, binding site is round. That fit will be perfect. If this one goes here or here, will not fit perfectly. Make sense? So this is the perfect binding site for this signal or chemical molecule, and on this integral protein you have that. So when that binding will be perfect, like you are putting your key in the lock. You all have cars here, right? Car, and you have a key. If you go to another car, you won't be able to start. Make sense? Must fit perfectly, right? To start. So exactly same thing. When that fit will be perfect, like key lock type fit, then the signal go to the inner part of the cell. This is inside the cell, this is outside of the cell, right? Chemicals are coming from outside. And that signal will start the intracellular machinery. For example, if this chemical is to produce a protein, then it will send signal to inside the cell, and signal will go to the ribosomes, right? And tell ribosomes that you produce this kind of protein through the DNA, of course. So just know that once the signal will go in, get in, then the cell will start to function as direction it receives. Okay, so that's how some integral proteins work as receptors. Some integral proteins work as enzymes. That means convert one type of chemicals to another through chemical reactions, by helping the chemical reactions inside the cell. So that's what they have shown here. One type of molecule will be converted to another through chemical reactions, and those reactions will be helped by some integral proteins. Peripheral proteins, I mentioned two functions of peripheral proteins. Intercellular junctions, also called intercellular joinings or membrane junctions. This. So, intercellular junctions hold the cells mainly together. So the cells can work together. Now, three types of junctions are mostly present in between the cells of our body. What are those? Tight junctions, desmosomes, and gap junctions. Tight junctions hold the cell membranes together, and they're very small, very tiny pores are present in tight junctions. So only very small molecules can pass through the tight junction. So mostly sealed, doesn't allow most of the fluid and molecules, prevent the fluids and molecules from moving between the cells. Only very small molecules can pass. Desmosomes. Desmosomes are very strong junctions. They hold the cell membranes together very strongly. So desmosomes are mainly to hold the cells together very strongly. desmosomes do what? Form the junctions by making zipper-like attachment. You know, in your clothes, you sometimes use zipper, right? You know, zipper you use in your clothes. So, desmosomes form zipper-like attachment. So, very strongly hold the cell membranes together. Gap junctions. Gap junctions have holes or pores to them so that molecules can easily move from one cell to the next, because gap junctions have many pores or channels. So here you see how the tight junction looks like. You see two tight junction is like that. This is one cell membrane, this is another cell membrane. They are attached to each other, and now if you stitch them together, stitching, okay, that's how the tight junction is. So two cell membranes are stitched together. By what? Interlocking junctional proteins. So peripheral proteins. Remember, all those are peripheral proteins. Interlocking junctional proteins. Desmosome, as I have mentioned, what happens? This is one cell membrane. This is another cell membrane. In the inner surface of the cell membrane, you have a disc-like structure. That is called plaque. So these two are plaques. Like, you know, a flat disc is attached to the inner side of the cell membrane, and from those plaques, linker glycoproteins arise, and these linker glycoproteins are also called cadherins. They do what? Form like this. So this is the plaque and linker glycoproteins, and hold each other like this. So very strong attachment is created between the cells. Gap junctions, you see, through the gap junctions you have channels or pores. So fluid can or ions can easily move from one cell to the next. So the cells can talk to each other quickly, right? By exchanging ions, they can quickly communicate or talk to each other. So those are three important types of intercellular junctions produced by the peripheral proteins. Okay. How you know that in the tissue you have intercellular junctions and they stay together? When you get your skin dry, right? Sometimes you see you can peel the skin. Certain animals, creatures, or snakes, you know, snakes, when they remove the skin, the whole chunk of the skin comes off, right? So the cells are attached by desmosomes. We know that. So the cells are strongly attached to each other. In heart muscle, you have plenty of desmosomes, because heart muscle fibers must contact, all fibers must contact at the same time, right? So they are strongly attached to each other. They work together. Also in the heart muscle you have plenty of gap junctions at the same time. So they can talk to each other by exchanging the fluid, right? Ions very fast, because in heart muscle you have also plenty of gap junctions. So ions can move from one to another quickly. Okay, so intercellular junctions are given by the peripheral proteins of the cell membrane. Now we'll see the transport across the cell membrane. You know that cell membrane is a semi-permeable membrane. That means what? That means it is selectively permeable, partially permeable. It allows some chemicals but doesn't allow others. So through the cell membrane, transport occurs by two processes. One is called active transports, another is called the passive transport. Passive transports don't require ATP or energy, but active transports require ATP or energy. Active, it is telling you, right? Active. So you need the utilization of ATP or energy. Now, since passive transport doesn't require ATP, it can happen in living as well as non-living membranes because no ATP is required. But active transport only occurs in living cell membrane because ATP, we know that, is produced only in living cells. Metabolism, right? Through metabolism, ATP is produced only where? In living cells. So active transport can only occur in the living cell membrane. Passive transports could be different types. What are the important passive transports? Important passive transports are simple diffusion, facilitated diffusion, and osmosis. Facilitated diffusion could be two types, carrier mediated and channel mediated. Now first we'll see the simple diffusion and osmosis. So if this is a cell membrane, we know that cell membrane is a semi-permeable membrane, okay? And in one side you have the fluid that has water and glucose. For example, just to give you an example, in this side. In this side, you also have water and glucose. So in both sides, you have water and glucose. In this side, the concentration is high. Okay? This side is low concentration because more glucose is here. Make sense? So concentration is high. In this side, you have less glucose. Is it clear? So that concentration is low. You know that. Now, this membrane is semi-permeable. So it will allow the water and glucose to pass through it. Glucose, since the concentration of glucose is high in this side, glucose will move from high to low. Make sense? From high concentration to low concentration. That is called simple diffusion. Simple diffusion. Okay. Okay. So simple diffusion moves the solutes. In this case, in this solution, solute is the glucose, solvent is the water. Water is solvent always, right? Water is solvent. So the solutes move from high to low. That is simple diffusion. Remember, always the solvent, in this case water, moves in opposite direction of solid. So this is water will move from low to high, and that is called osmosis. So movement of solvent or water from low to high, that is osmosis. So this is simple diffusion, this is osmosis. If you just think very simple, what's the goal? Anybody? Why the chemicals are moving, glucose and water, in opposite direction? To bring equilibrium, same concentration in both sides, right? That is the goal. Is it clear? So now you think, just forget everything. This side you have less glucose, right? This side you have less glucose. So glucose should move from here to here. Make sense? In this side you have more water, right? Less glucose, more water. So water will move from here to there, and always opposite. So very simple. The goal of purpose of movement is bringing equilibrium, same concentration in both sides. So if you just know that, then you can easily tell who will move in which direction. This one is more glucose in this side, so it will move in this side, and water will move to the other side. So simple diffusion and osmosis. So water moves from what? Low to high concentration. Is it clear? Water moves from low concentration to high concentration. That is osmosis. Now, facilitated diffusion. You remember, I told you, in the cell membrane you have integral proteins, right? And I also showed you that some integral proteins have channels, two channels. So those molecules are smaller than the size of the channel, they will pass through the channel. So that is channel mediated diffusion, going through the channel. Some integral proteins work as carrier. What happens? This is the integral protein. Inside the integral protein, you have a site, a seat for the molecules. So what will happen if this is a molecule that will get in, will go in and will sit here. You know, sometimes we go to the Six Flags and sit on the carrier, right? Sit on the carrier, and then what will happen? This integral protein will change the configuration, shape will be changed, and will drop it to inside. So first the chemicals will sit there, then the integral protein will change the shape, and then will drop it to inside. So that's how some integral proteins work as carrier. So these are passive transports. This is just an experiment you can do in the lab. You can put the solution, glucose solution or sugar solution of two different concentrations that I showed you here in a U-shaped test tube, and separate them by a semi-permeable membrane, and you will see the water will move from low to high and solutes will move from high to low until in both sides the concentration becomes same. Make sense? And when the concentration will be same, no movement. So that is one test you can do to see the movement of solids and water or solvent. Another thing you can do is put a membrane which has very small holes. Only will let the water molecules pass through it, but not the solids. In that case, what will happen if you put the solution of two different concentrations? You see the membrane, in this one, the hole is big, so water as well as the solids can move. So they will move in opposite direction. In this case, the holes are very small. Only water can move. So what will happen? Water will move from low to high. We always know that water moves from low to high, right? The goal is what? Bringing same concentration, right? So water will keep moving until the concentration becomes same in the other side. Same in the other side. So what will happen in this case? Since solid cannot move, only water will move from low to high. And the concentration will be same on both sides, but the volume will be higher in one side because water has moved from low to high. Okay, so here you only see osmosis. Now, how a living cell responds in different types of solution. If you put a living cell in an isotonic, hypotonic and hypertonic solution, how the living cell will respond. So now just think that you have a beaker and the solution here, the concentration is 5 milliosmol. That's the unit. And if you drop a red blood cell, for example, inside the cell, the concentration of fluid is 10 milliosmol. 10 milliosmol. So this solution is hypo or hyper, you know, compared to this. Everybody. Compared to this solution is what? Hypo or hyper? Hypo. Hypo means what? Lower, right? Less. So this is 5, so this is hypo. Make sense? Is it difficult? Yes or no?
So that's just the fluid?
Fluid, comparing. You are just simply comparing.
So
this is 10.
Because it has more water. So that's why
Don't think about that. Just I have asked. Here the concentration is 10. Here the concentration is what? 5. Make sense? So this solution is hypo compared to this. Make sense? Now if I ask you in opposite way, inside the cell, the fluid concentration is hyper or hypo compared to outside? Hyper. Is it clear? Now you are putting here a cell in hypotonic solution. Is it clear? This solution is hypotonic. So hypotonic means the concentration is low. That means you have more water here, and water will move from here to here, because water always moves from low to high. That is osmosis, right? I had mentioned just a few minutes ago. So water will move from low to high. Now when water gets in, the size of the cell will get what? Bigger. Make sense? Because water is getting in, right? Gradually it will get bigger and bigger, and even it may rupture. If the cell membrane is weak, it will burst or rupture. So that is the response of a living cell in hypotonic solution. Is it clear? So this is hypo. The cell will get bigger and may rupture, swelling of the cell. Now, if I change this solution, now the concentration of solution here is 20. So compared to the cell, this is what? Hyper, right? This is hyper, more, double. Always water will move from what? Low to high. So in this case, low to high, that means water will leave the cell, right? Will get out from the cell. So what will happen? The cell will get smaller, shrink, right? The cell will shrink. That is the response of the cell in hypertonic solution. Water will get out. Is it clear? Now if you put in isotonic, that means iso means what? Equal, same, right? No movement of water. So the cell size and shape will remain same, will not change. Is it clear? So which one you will prefer if you inject saline or fluid in the vein? Which one you will prefer? Iso, hypo or hyper? Iso. Definitely iso, right? It will not bother the cells. It will not change the shape or size of the cells, right? If you inject hyper, then fluid will come out from the cells, right? If you put hypo, fluid will get into the cell and may rupture the cell. Is it clear? So you will prefer what? Say everybody. Isotonic, right? So that is important to know. Okay, so we'll start.